Hello, my name is Cyril Jacob Joseph, and the date today is December 5th, 2023. This is the DNA Extraction Lab.
So I've gathered all of my materials here for this lab. And now we're gonna take this blender right here, and we're gonna add our split pea packet into the blender, as such. Then we're gonna add salt packets.
At step 1, sodium chloride is added to break down the cell membranes of the split peas. This helps release the DNA trapped inside the cells.
Then we're gonna add 1 cup of measured cold water, and we're gonna add it into the blender. Then on the high setting, we're going to turn the blender on. And then after 30 seconds, we just stop the blender. Now, we've placed a cheesecloth over a drinking glass, and now we're going to pour the contents of the blender through the cheesecloth into the glass, to this cheesecloth. And now we're going to carefully remove the cheesecloth from our glass container, and actually we can go ahead and throw away this cheesecloth and the contents of it into a trash can. Then we're going to add our detergent liquid into the solution.
At step 6, detergent is added to the solution to break down the cell membranes and disrupt the lipid molecules, allowing the DNA to be released and isolated.
I'm going to gently stir with my glass stirring rod with the content of this. And now after stirring, we're going to put it in the fridge for 10 minutes. Okay, it's been 10 minutes now, and we're going to remove the glass from the refrigeration. Okay, now we're going to add the chilled contents into this glass tube and fill it up to about 1/3. And now we're going to add approximately half of our meat tenderizing powder into the glass. And now we're going to stir the contents gently.
Papain powder is added to the solution to further break down proteins and cellular debris, which can interfere with the DNA extraction process.
Holding the glass tube at a 45-degree angle, I'm going to pour the contents of isopropyl alcohol into the glass tube until they're equal amounts of alcohol in solution.
At step 14, chilled alcohol is used to precipitate the DNA. When cold alcohol is added, it causes the DNA to come out of the solution and form visible strands, making it easier to isolate.
Okay, now we're going to hold this upright in an upright position for a minute. Okay, and now we are observing the interface between the two layers. And as we can actually see, we see the DNA at the interface between those two layers. And my observations are that the color is white, almost clearish in nature. It is more than I expected, the amount-wise, and it is more viscous than I expected. And it's almost clumping together, even especially at the top of that matter.